The moment a hair follicle leaves the scalp, the clock starts ticking. But what exactly keeps these microscopic organs alive while they wait to be implanted? Once extracted, a hair follicular unit becomes a living microorgan entirely cut off from its blood supply. To survive, it must rapidly consume stored energy, primarily in the form of ATP. To slow this metabolic drain, the tissue is immediately placed in a temperature-controlled physiological saline solution. From a planning perspective, Dr. Erkam emphasizes that minimizing the out-of-body time is just as critical as the extraction process itself. This isotonic fluid prevents cellular dehydration, while chilled temperatures around 4 degrees Celsius significantly reduce the rate of energy depletion and oxidative stress. In his clinical approach, Dr. Erkam insists on strict protocols for holding solutions to protect the delicate dermal papilla cells. Keeping the tissue fully submerged prevents desiccation, which is a primary cause of cell death during this brief waiting period. In practice at Hair Upload Clinic, Dr. Erkam notes that careful temperature regulation bridges the biological gap until the tissue reconnects to blood vessels. Evidence from transplant guidelines shows that proper storage medium and thermal control consistently correlate with high tissue viability. Ultimately, understanding this holding phase reveals why controlled storage environments are fundamental to successful transplantation biology.